Kim Baumann. I'm Chief Editor of Nature Review's Molecular Cell Biology, and I'm here with Aaron Straight, who is Professor of Biochemistry at Stanford. Hello, Aaron.
Thanks for taking the time to have a conversation today.
So your laboratory works on different aspects of chromosome biology that are related to understanding how the segregation of chromosomes occurs, so that in a correct way. And this includes work on the centromere and how it drives the assembly of kinetochores, as well as on RNA biology and how RNAs regulate the structure and organization of chromosomes. So could you start by telling us a little bit about the connections between these two fields, and also if that's something what excites you particularly at the moment?
Yeah, actually that's a good question, because it's not, those can seem like somewhat disparate fields and not necessarily connected, but maybe just historically how I ended up in trying to understand chromosome segregation. I started out just because I was interested in the mechanics of chromosome movement and how, what controls the dynamic movement of chromosomes during anaphase and spindle elongation. And those problems are still interesting problems. In some ways, though, I feel like we've gotten away from that because something else sort of caught my attention, which I think is a really fascinating problem in cell biology that will take decades to play out. And that is that both of those examples you gave, which are near and dear to my heart, I think are not so much about the specific mechanics of moving chromosomes and controlling the regulation of when they separate, but more about how a cell sets up specific functional domains of chromosomes. And in the face of all of the other cellular processes, whether that's mitosis or differentiation, how it decides how to specify a particular function and then attempt to reinforce that function over time, whether it's on a short time scale like a cell cycle or over the lifetime of an organism. And so let's see how to bring those two aspects together. I think one way to view it is through the lens of what I work on for the most part is chromosome-based functionalization. And the centromere is a great example of that because it's made of DNA and histones and chromosome proteins, but it has a very unique function that's distinct from the content of the other parts of the chromosome. And that function, as far as we know, is, at least in humans, primarily encoded not at the DNA sequence level, but by the proteins that associate there. So that sets up an interesting problem.
And it's a function that is also maintained as the cell divides. It's spatially in the chromosome. It's a region of the chromosome that is.
So that gets right to the heart of it, right? Because if it's within some part of the chromosome and it's not the sequence defining it, then what gives it boundaries and what gives it identity? And as you said, as the cell is going through division, if the cell is duplicating and diluting it every time it replicates its DNA and chromatin, how is it sort of maintained or reassessed? And so we spent several years trying to understand that problem. And it's really the core of that problem for the centromere is that there's a specific histone that's distinct from the rest of the histones. And so there must be some cellular mechanism that can recognize that and say, this is where we put more of this identifying histone, and we don't put it elsewhere, and we don't put other things here that shouldn't be here. And so even though that sounds trivial, it turns out to be quite a complicated problem to solve. And I think we're starting to touch on the machinery that does that, that sort of recognition of what a chromosome does. I think we're far from understanding how you couple that recognition to the reassembly and the protection of that part of the chromosome from the rest of the chromosome.
And so how does RNA fit into this?
So that's the RNA, we're very new to that world. So we've really been working on that problem seriously for maybe 4 or 5 years. I guess that's new, I don't know.
I guess so, yes.
But, and we fell into that almost because of an accidental study of the centromere region. And we were interested in the general concept, which is there are certain RNAs that are well established that bind chromosomes and change their function. And the best is in female dosage compensation. One X chromosome gets coded by an RNA and silenced, and the other doesn't. And so we sort of asked the generic question, are there other RNAs that might act in that kind of a mechanism to control the chromosome fate or chromosome structure.
And do you mean specifically to have regions that are heterochromatic regions or not, or beyond that, or more than that?
I wish we'd been that well informed when we started, but I don't think we had really thought through the problem very well at that point. So we just took an unbiased look. What RNAs stick to chromosomes? But we did that in mitosis when transcription is for the most part repressed.
And so is this in which cells? Which system is this?
Yeah, this is in human cells.
Human
so
cells,
we jump between a lot of different organisms in the lab, but this RNA stuff has been in human cells, though we are now moving it to other systems. But the idea was, can we take an unbiased look? Can we just essentially take a census or a visual assay to ask where do RNAs bind on human chromosomes? And we chose mitotic chromosomes because transcription shut down for the most part. So we did that. We just developed a labeling system to look at it. And we saw something pretty extraordinary, which were RNAs stayed associated with mitotic chromosomes. And in particular, they concentrated around centromeres, which we didn't anticipate that would be the case, but we knew how to work with that part of the chromosome, so that helped get us more interested in the problem. So we've spent a few years now working on that, and we've begun to understand that this,
So are these RNAs that are transcribed from those regions, and then they remain there? Or they associate there after they've, how does it work?
That's actually an extremely good question, because they're exactly as you just described it. They're RNAs that are transcribed from those regions, but maintain their association with those regions. And we've recently discovered that those RNAs are involved in bringing a series of histone-modifying enzymes to that locus to actually modify the chromatin and help repress it. So that's how it comes back to the centromere, right? Because these are, in some sense, non-sequence dependent events, right? We don't think the RNA sequence is important.
I was going to ask, so is there any structure, specific structure perhaps of the RNAs? Or are there characteristics of these RNAs that make it
once again,
work the way they work?
Yeah, that's a great question. We have tried very hard to understand that. We have even done some specific structure mapping on the RNAs. We really have no evidence that there's a specific structure involved. Instead, I think what we're thinking is that the RNAs are acting as a platform just to increase the local concentration of the enzymes that bind them. And that opens up a problem.
So it's mostly, say, a physical function.
We think so.
Is there any regulatory function beyond, because it brings enzymes on, right? Like that modify. So is there also a way, do they regulate also the activity of the enzymes, or is it only just bringing
good question.
over the
We have tested that, and our data so far is that there's not specific regulation of the enzymes. And so what we've kind of come to is that rather than changing the activity, they essentially localize and concentrate the activity. And so it's a way of redistributing the activity to a particular site as opposed to modifying an activity so it acts in one place and not another. And that, coming back to that larger problem, how these are related, I think both of those problems are problems in, how do you maintain an epigenetically defined locus? And so the RNA, I would say the RNA, project that we've started to work on is really still in its infancy. And I think that's a general problem for people studying non-coding RNAs in any organism, in that now there are tens of thousands identified, and we know the functions of a handful.
Oh, very few. And do you know anything also on how they are activated? I mean, are they constitutively reactive? Because these regions are defined and remain there, right? I mean, this is one that is, a region that is maintained over. So are they constantly being produced?
So we've looked at that, and we know that they're being synthesized in one particular part of the cell cycle in G2 before the cell goes into mitosis. And maybe that's just to make sure that the transcript is there, so that when the chromosomes condense, it's at the right place at the right time. We don't know what the regulation of their synthesis is. It's too early, and we just haven't done the experiments. But that's an interesting problem, whether there is some regulated behavior that allows a cell. For instance, if you go to an early embryo or a pluripotent cell, they lose a lot of that heterochromatin. And so there has to be some regulation of that domain, whether it's through the RNA or whether it's through something else.
And do they function also in other parts of the chromosome? Are they specific to centromere?
They're not specific to the centromere.
So
and
it's
we're
maintaining very different
Yeah,
regions, let's say, or defining perhaps
we
different
think
regions?
so. We think for this particular class and this enzyme, these modifying enzymes, that it's really involved in repressing all kinds of different repeat sequences in the human genome, whether those are centromeric, whether they're transposable elements, or endogenous retroviruses. Whereas we don't think these RNAs affect sort of the euchromatic genome very much. We think this process is quite specific to making heterochromatin and keeping it.
Yeah. So what is the next step in studying these and other aspects that you're
I mean, the
really
thing
interested?
that I would say I'm probably most excited about at the moment is, so we realized that we were studying one RNA, right? We just had this one thing, and that's essentially how the field has been going. You'll find an RNA that's interesting, and you'll bang away on it for a while and learn something about the biology of that one RNA. But what we wanted to do is ask, can we develop a method that allows us to look at all the RNAs, right? Can we globally find all the RNAs that bind to human chromosomes and then understand where they're acting and maybe try to understand how they're functioning? And so just over the past 2 years, we've developed a method called chromosome-associated RNA sequencing that allows us to essentially link an RNA to the site where it's bound to the DNA through a direct linkage between the RNA and DNA.
Okay.
So that means regardless
so it's a spatial, it allows you to see exactly where they are
Exactly,
located,
yeah,
in
and it
terms
makes a
of
physical link between the RNA and DNA. And that's advantageous because it doesn't matter if it's coming from the site where it's transcribed or if it's going to some other part of the chromosome and acting, as long as we can capture that interaction. And so we've just recently done that and shown that we can then, using high throughput sequencing, identify all of the RNAs and all of the sites where they're bound. And so by doing that, you immediately begin to generate hypotheses about how an RNA might bind. So for instance, we developed this technique in Drosophila because the genome is a bit smaller, so it's easier to do. And the first thing that just jumped out of us is the dosage compensation RNAs, which control the expression of the X and Y balance, just beautifully coat the entire X chromosome. And so we know that we can detect cis interactions for those RNAs, but we also see those RNAs going to other parts of the genome, and we see a lot of other RNAs with interesting behaviors. And so I feel like this is just beginning to scratch the surface. And
it would be also interesting to see how they are targeted, I guess, to certain regions. It's a bit of a chicken and egg thing, because they target factors to the chromatin and vice versa. I guess if they are not just where they are transcribed, they need to reach.
Yeah, so I think that
destination.
question is one of the most important questions in the field right now. Because for non-coding RNAs, I would say for most of them, we do not understand how they're targeted to where they act, and we don't understand how they're maintained there. So, we know that RNAs, they can bind different, complexes and RNA binding proteins, but what gives them selectivity for a place in the genome is really a black box. And so if we can tackle that question over the next decade, that's a really great problem. And I don't think that's not a problem that's going to be solved by one lab.
No, of course.
That's a problem for the whole field to try to understand.
Well, I guess there can be mechanisms that involve sequence-specific and non-sequence specific events, right?
Absolutely.
With many factors, perhaps.
Yeah, and there are good examples of sequence-specific and non-sequence-specific factors that are involved in the process, but I think we still don't understand the specificity problem, right? How if it's sequence-specific, we do understand it to some extent,
a little bit,
right?
Yes.
But for the ones that aren't, that's a much tougher problem. And so it's fun to think about.
And just going back to the RNAs that are responsible, so they function in the centromeric area, just as. So I'm thinking, well, just how they are kept there and how they. If you say that there's no sequence specificity, but so what does it tell you how that region functions, and also what is, say, if you were disrupting any of those mechanisms, what happens to start with? Is it that just
no? This is a very good question. I guess one way to view this, the poster child for understanding these problems are really studies in fission yeast, and that arguably is where mechanisms for RNAs controlling chromatin have been best worked out in these regions around centromeres. In that case, though, it's known that there is sequence-dependent interaction that help control, shutting down the chromatin. And if you destroy that system in fission yeast, it causes chromosome segregation. It de-represses.
It does. Then the whole
so we've
machinery
just,
is disrupted.
Exactly.
Then, yes.
So we've just recently been able to test that in humans, and it really is because now that you can do things like modify genes with CRISPR and actually make mutants in the genome like you would in yeast, now you can do those experiments. And so what we find is when we compromise that RNA-dependent system, we start to de-repress all of these heterochromatic regions that depend on repeat sequence. And so that's part of why we think one of the key features of these systems is to ensure that you maintain repression of heterochromatic regions, which is a bit chicken and the egg, right?
Yes.
Because it requires transcription in order to repress the transcription that's occurring there. And understanding how that balance plays out, whether that's a way for the cell to be able to regulate the relative expression from some of these regions, is also an interesting problem to think about. Yeah.
Right. Well, so I guess this is going to be a major focus for your lab. Will it be
Yeah,
a major focus for your lab for now? Because you say it's reasonably new, just a few years. Is it something that is going to be?
No, we'll keep pushing on this for
lab?
a while. Yeah. I mean, we haven't abandoned the centromere. I love that. I love that part of the chromosome, and we'll keep working there. But I think this represents maybe a broader class of problems, and hopeful that we'll get some interesting insight.
All right. Well, thank you for talking to
me.
Thank you for your time, and I hope you enjoy the rest of the conference.
Yeah, you too. Thanks a lot.
Thank you.